Good morning, everyone. Thank you very much to BRIC for organizing this little webinar. So my name is Guilhem Tourniaire, and I'm going to present you the technology we have developed for single cell and organoid sorting and isolation using image-based dispensing technologies. First, a few words about who we are. So we are Cellenion. Cellenion is part of the BICO group, and BICO is an international group of companies composed of 15 companies. And our goal is to develop new technology to revolutionize biotechnology in general. And we call this the bioconvergence concept. The mission of Cellenion is to provide disruptive technologies, products, and services to better understand and master cell biology. And I'm going to give you more detail about the technology we have developed at Cellenion over the years. So first, I will start with a short introduction on the concept of image-based single particle dispensing. Then I will introduce you to the two devices that we have developed, the CellenONE for single cell isolation and the spheroONE for spheroid and organoid sorting and isolation. And we will finish by a question and answer session. And what we're going to do is we're going to keep only the droplets. We're going to generate small droplets, and we will keep only the droplets containing a single cell. We will discard droplets that are empty. We will discard droplets that contain multiple cells, only to keep the droplets that have a single cell. And it's an image-based technology, so we can sort cell also based on morphology, but we also have like fluorescence module allowing us to sort cells based on fluorescent marker, up to 4 markers. So I have this little animation that's going to show you how it works. So we start always from an aspiration of a cell suspension into the dispenser. The dispenser is going to be positioned in front of a camera. It's kind of a microscope. Then we look through the dispenser, 2 modes of visualization, one which is equivalent to a bright field microscope, one to a fluorescent microscope. At the tip of the dispenser, we have this ejection zone, which is the equivalent of the volume of 1 droplet. And this sedimentation zone that is there to account for the fact that cells sediment when there is no action. We generate small droplets. Every time we generate a droplet, we take an image of the tip of the dispenser. If we have a single cell in the ejection zone, we know that in the next droplet we will eject, that single cell will be ejected. We go back to the camera, image again, another single cell. Second position of the target, typically a microplate or microchip. Obviously, not all of the droplets are going to contain single cells. Some will be empty, some will be containing multiple cells. In those cases, we discard those cells. Particularity of our technology is when we discard, we put that into a small tube. So if you're working with a very scarce sample, a sample containing very few cells, then you have the ability to reprocess this sample, not to lose any of the cells. Being an image-based technology, we take a number of measurements on the cells. We measure the diameter, the elongation, and the circularity. And this allows us to differentiate a single cell from a cell aggregate, for example, 2 cells bound together, or debris. And by doing so, we make sure that we only isolate the cell of interest. In terms of speed, we can do this isolation at a speed up to 40 single cell isolated every minute. So that was an animation. It's nice to explain how it works. Now, in real, in terms of, I now have like a video that is going to show you inside the device what is happening. So here I have the dispenser, my camera. Here we are isolating ELA cells into a 96 well plate, and at the bottom is what's happening in the software. So what you have to consider is that the image of the dispenser has been tilted 90 degrees clockwise. So this is the tip of the dispenser, my boundary of the ejection zone, my sedimentation zone. Whenever I'm going to have a single cell in here on my image, I know that in the next droplet, I will isolate a single cell. So I'm now going to start the video, and you will understand better how the system works. So I have a single cell in the ejection zone, again, a single cell, single cell, single cell. Sometime you will have maybe a doublet. You're going to see soon, and we will pause the image. Here we have a doublet, so we paused so that you have time to see it. So it's detected as a red circle, as you can see, outside of the fitting parameter. Again, some very large cells that were outside of the fitting parameter and not isolated. So as you can see, it's very quick and straightforward. So using this technology, we have developed 2 systems. One that was released in 2017, which is called CellenONE. It is mostly used for isolation of single cell. These cells can be of any size from 0.5 micron all the way up to 80 micron. So we are able to isolate bacteria, other microorganisms, spores, but also all type of mammalian cells, up to very large cells, liver cells, and even cardiomyocytes. More recently, last year, we released a new device, which we call the spheroONE, which works on the same principle, but that is dedicated for the isolation of larger particles, ranging from 50 micron up to 600 micron, and we use this for isolation and for sorting and isolation of spheroids and organoids. And I will tell you more about this at the end of my presentation. But first, I will introduce you to the CellenONE technology. So inside the CellenONE, it's we say it's an all-in-one system. You can do single cell isolation, but being based on a dispensing technology, we can also do picoliter to nanoliter dispensing. The droplet we are generating range from about 50 picoliter to 800 picoliter in volume. And we also have a system that allows us to do heating of the target plate, and we control the humidity, which is very important when you're working with such low volume to prevent evaporation. In terms of application, this device is very versatile. We are addressing multiple markets in the field of single cell analysis, single cell omics, and also for automated cell line development. So the technology is based on the piezoacoustic technology. We have a glass capillary, a piezoceramic here. We send an electrical pulse to the piezoceramic. This generates an acoustic wave inside the glass capillary. And for every pulse that we send, we generate a small droplet. What has allowed us to do this single cell isolation is the fact that this technology is very reproducible. Every time we send the same pulse, we will get the same droplet. So then the ejection zone is always constant. It's very precise and very accurate. And it's also compatible with a range of different solutions, both aqueous solution, but also organic, which comes handy for certain applications such as single cell proteomic sample preparation. We can handle any type of sample. We can aspirate sample as small as 1 microliter containing just one cell. Obviously, we can do hundreds and thousands of cells as well. As I said, we are very versatile in terms of cell size. The speed is about 40 cells per minute. We can reach up to 100% single cell accuracy, meaning that every of your wells will contain a single cell and only a single cell. Being an image-based technology, we have an image of every cell that is isolated. We know in which well these have been isolated, and this allows you to link the morphological parameter of the cells together with the omics data that you will be generated. Or we can use this as a quality control to make sure that only single cells have been isolated. As I said, application in the field of single cell omics, these range from transcriptomics to proteomics to genomic. And in terms of cell line development, we address the market of immunotherapy, gene editing, and stem cell development. So I will now focus and give you some example of the benefit of this technology in the field of automated cell line development. So as I said, for monoclonal antibody after CRISPR-Cas9, gene editing, or for people doing research in stem cells. So typically, the traditional way of obtaining a single clone was to undertake serial dilution. I imagine most of you have gone through that in your research. It's very tedious. You have to do multiple rounds of serial dilution in order to ensure that you have a single cell at the end in your well. What we offer with the CellenONE is to drastically reduce the time to obtain those single clone by automating this process. These are some examples of data generated by cloning a CHO cell line, which is widely used in bioproduction. Here we compare the results that we obtain in different microtiter plates. We did the experiment in 96, in 384, and in 1536 well plates. And as you can see, here we are measuring the clonal outgrowth. What is the clonal outgrowth? Clonal outgrowth is a combination of the accuracy, making sure you have a single cell, but also making sure that this cell is going to give rise to a single colony. So here we are measuring the percentage of wells containing a single colony coming from a single cell after 7 days of culture. And as you can see, we obtain outstanding results because the technology is very, very gentle. Unlike FACS, we don't put any stress on the cells because we are using this acoustic wave for generation of the droplets. And this ensures that you maintain very high viability of your clone. On average, 95% clonal outgrowth. If you were to do that by manual pipetting, at best, you would have 25% of well containing single cell, but within those wells, you will probably have some wells containing multiple cells. At some large pharmaceutical company that were using the FACSARIA, they asked us to compare our device with the FACSARIA 3 that they were using. And in this experiment, they supplied us with samples containing exactly 1,000 hybridoma cells. The goal of this experiment was to compare those two devices. And the first measurement that we did is how many wells were cloned, how many cells were cloned out of the 1,000 cells. So this is what we call the cell recovery. So out of 1,000 hybridoma cells, with the FACSARIA, 533 wells were cloned. So they tried to put a single cell in 533 of those wells. With the CellenONE, it was slightly better, with 664 wells. But where the most striking results is in terms of the clonal outgrowth. So again, linked to the viability of the cells after isolation. And there you can see a massive difference. Out of those 533 wells, only 75 wells contain a single colony using the FACSARIA. With our device, the CellenONE, 480 wells contain single cells. As you can see, this is much more gentle than a FACS and much more suited for automated cloning. We took this to the next level, actually, by completely automating the process. And that's a new device that we released last year. And we have now installed at two that we have now installed at two big pharma company. Sorry, I'm having issue with my automation. Sorry, here we are. So here we see the side of an automated incubator. For the one you know, it's a Lyconic incubator, but this can be done also with a cytomat from Thermo Fisher. So in this platform that we have developed together with our sister company BioZero, we have an automated arm that takes the plates out of the automated incubator, bringing to the side of the CellenONE HT. So this is our device, the CellenONE HT. We undertake an anti-static charge removal. So this is to ensure higher accuracy of our cloning. Then we undertake the isolation of 384 clone into those plates, and the plate is ejected. The lead is placed back. Then we take the plate. In this platform, one of the two platforms that we have supplied, we have an automated centrifuge. We do a quick spin with the centrifuge, and then we place the plate back into the incubator. This is plate number 1. Then we repeat the process with how many plates as you want. And this is completely autonomous. We don't need any operator in front of the system. So our customer, what they do is they prepare their sample during the day. And then in the evening, they start the experiment. And they are able to clone thousands of clones completely autonomously. So this is what the platform looks like. Robotic arm, automated incubator, the CellenONE HT, and an automated centrifuge. This is compatible with any type of format, 96, 384, 1536 OL plate. And as I said, yeah, we've installed 2 of those platforms, and our customers are really happy with the performance we are able to achieve. To give you an example, with one of our customers, we are cloning 8 times 384 wells per sample, and they typically run 4 samples per night. This gives us, in the morning, 12,000 clones completely automatically and autonomously. So you don't need any operator to undertake that. So this is really a game changer. In terms of what technology they use, they replace with our technology. In one of the customers, they replace the Beckman Coulter MoFlow system, and the other customer replaced the Berkeley Light beacon system, which they were not really happy about the performance of. I will now move on to the field of single-cell omics. What do we do with our technology in the field of single-cell omics? So for the one of you who are familiar with single-cell analysis, you will have probably seen this little representation of what are the benefits of a single-cell analysis. So we take this example of the smoothie and the fruit salad, if you want. In a smoothie, everything is mashed up. So it's very difficult for you to identify all the fruits that are contained in the smoothie if you want. If you look at a fruit salad, you can see every single fruit, and then you can better analyze, for example, a very small population of fruit that is present, but in very scarce number. And this brings a much more detailed picture of what is present in your tissue. So in a human tissue, you know you have a very heterogeneous population typically, and you want to identify certain subpopulations that are going to have a very important biological role. And that's exactly what the field of single cell analysis allow you to do. By analyzing every single cell, you can better understand what is happening in the tissue, in the body of someone. First example of work that was developed on our CellenONE platform at the University of British Columbia and the Memorial Sloan Kettering Cancer Center in New York. It's a technology, a method that is called Direct Library Preparation Plus. It was published in Cell in 2019, and they recently published a very nice Nature paper in 2021 using this technology to analyze cancer tissue. It's a whole genome sequencing method that is now implemented in multiple samples around the world at the Crick Institute in London, MSKCC New York Genome Foundation Center, at the Japan Foundation for Cancer Research, and the Cancer Children Hospital in Sydney. The technology allows you to miniaturize your whole genome sequencing sample preparation. And it allows you to do a number of measurements. You can do copy number variation, you can do clustering, single nucleotide variation, and reconstruct lineages. I really encourage you to go and look for this paper. It's very, very nice. So this customer developed their own chip and their own protocol. So a few years back at Cellenion, we decided to make the best of the capacity of our device by developing our own consumable. And what we did is we developed those microscope slide format nanowells. So these are little slides containing 384 wells in which we're going to undertake single cell isolation and library preparation, part of the library preparation. So this allows us to miniaturize our workflow, reduce costs, and improve sensitivity. Working volume in those is between 20 and 500 nanoliter. So here you can see we have developed also a range of holders that allow us to place those in our device, in centrifuge, and so on. And this is the different steps that are involved in the first kit that we have released. We have also developed a chemistry allowing you to do single cell transcriptomic analysis on single cell. So the chip that we are supplying in the kit arrives already pre-barcoded. So each well will contain a unique barcode, oligo-DT barcodes, and they're also pre-filled with a lysis and RT buffer. This is to make your life easy, basically. You receive the kit, you place your chip in your freezer, and you keep them there until you are ready to do your single cell isolation and single cell analysis. So on the day of the experiment, you just take the chip out of the freezer, place it into the CellenONE, and what you do your single cell isolation. You isolate your cell, you drop that into the lysis and RT buffer. So this represents a droplet of about 300 picoliter. In the well, each well contains 100 nanoliter of reaction volume. And then you take your chip and place it into a thermocycler to do your reverse transcription at 42 degrees for an hour and a half. Once you've done that, we have developed a small funnel device that comes on top of the chip, and you spin that into a centrifuge. You collect the volume contained in all of the well, and thus you collect your cDNA, and then you carry on your library preparation in a traditional manner. With this little kit, we did some experiments. So this is one of the first experiments we did, is we compared the same cell line grown in 2D versus 3D. This is the RG cells. They are widely used in the pharmaceutical industry for hepatic toxicity studies. And there we wanted to see if we use even a very low number of cells analyzed. In this experiment, there were about 100 cells for each condition, 2D versus 3D. And we wanted to see if we could see differences in gene expression. And as you can see, we noticed a lot of differences, and we were able to confirm that obviously the cells that were cultured as spheroids were much more similar to liver tissues with high expression of albumin and many other markers like gamma fibrinogen and so on. So now it's commercially available. It's a complete solution. You can buy those kits that works with our CellenONE device. This is a complete solution for 3' single cell RNA-seq preparation. It works in ultra low volume, so it's very sensitive. It provides very high quality data, and it's ideal for sample containing low number of cells. You can have up to 3,000 cells per experiment if you put 8 of those chips on the deck of the CellenONE. I will now move to another field that is actually very promising. We believe it's one of the future in biology, is the field of single-cell proteomics. And I will give you just a short introduction explaining what are the challenges in this field. So as you know, in genomic and transcriptomic, we benefit from the presence of PCR. We can amplify any oligos can be amplified. So we don't have so much of issues with sensitivity and presence of very low material in the starting sample. In the proteomic field, it's not possible to amplify proteins. So we have to work with whatever is present in a single cell. So it is really critical that during your sample preparation, prior to your mass spectrometry analysis, you're not going to lose anything from your sample. And also you have to work with very high sensitivity mass spectrometer. The approach we are using is called bottom-up or shotgun proteomics. We take the cells, we lyse the cells, we obtain the proteins, then we digest the protein to obtain peptides, and those peptides are injected into a liquid chromatography that is connected to a MS system. So the peptide fragments are going to get fragmented, and then the fragment will get again fragmented. And then using bioinformatics, we are able to reconstitute to detect and quantify the presence of the different peptides and proteins that were present in the sample. The field of single-cell proteomics is a very young field. It's been around for about 5 years. And it's thanks to the improvement in the sensitivity of mass spectrometer. And there are 2 main methods on the market today that are competing. There is a label-free method, which is using, for example, the Bruker timsTOF SCP. It has easier sample preparation. It provides absolute quantification, but it has very low throughput. We analyze 1 cell at a time. And currently with the timsTOF SCP, for each cell, it takes about 20 minutes of analysis. Then there is a method that is better suited to the Orbitrap technology from Thermo Fisher, which allow you to multiplex samples. So you put a little tag, a label on the peptide, and this allows you to pool multiple cells together. You can identify more protein with this method, but it's more complex to prepare the sample. It uses Chi channel, which is a sample, one well, which will typically contain hundreds or 200 cells. And it provides a relative quantification. And I will show you what we can do with the CellenONE in both those sample prep. So these are the different steps involved in the methods. So in the label-free, it's very straightforward. You isolate your single cell, you lyse it, you undertake the digestion with trypsin typically, and then you inject into your mass spectrometer. For the multiplex approach, you have to add a tag. So you do first your isolation, your lysis, digestion, that's similar. Then you go on and do your labeling. So each cell will be in a well, and you will add this label in each of the well. Currently, this technology allows you to multiplex up to 18 single cells using TMT isobaric tag. Then you have to quench the reaction. Then you can pull the sample because all the peptides are labeled, and then you inject that into your Orbitrap. And this is the chip, the first nano chip that we developed together with the Vienna Biocenter. It allows you to fully automate the multiplex approach. So this little video is going to show you the different steps. So we have developed those chips. They are made of Teflon to prevent any adhesion of peptide onto the surface. We can place up to 3 chips on the deck of our CellenONE. Each of those contain an array of 16 wells. So there are 12 arrays on each chip of 16 wells. First, we add the lysis and digestion buffer in each of the wells using the CellenONE. Then we undertake single cell isolation in each of those wells that has been pre-filled with a reaction mix. We then undertake the incubation on the deck. We have the ability to heat up the deck to 50, 60 degrees, maintain it at this temperature for a given duration, typically about 1 hour. Then we're going to aspirate the TMT label. So as I said, we have 16 different labels. We aspirate each of the label and going to place it in one of the well of each array. So this is for label number 1 that goes in the first well of each of the 12 arrays. We do the quenching and then we are ready to do the sample pooling. We do that by centrifugation. Again, we have those little funnels that are going to come on top of each array. We place that into this little centrifuge holder, then into the centrifuge, we give it a little spin. And basically what you do is you collect the volume, the liquid that is contained in those 16 wells into a small funnel. And then what we developed is this small little adapter, and this allows you to go directly into your mass spectrometer autosampler. By doing so, you don't have to use a single pipette throughout your experiment, because every time you're going to use a pipette, some of the peptides are going to be attaching to the cone, and you will lose some of the peptide present. So using this little kit that we developed, you are able to obtain really, really high quality data. So these are, again, the different steps that are involved there. And this is some example of data that have been published in a preprint on bioRxiv. The full paper is due to be published soon, hopefully in a very nice journal. And with this method, we are able to detect up to 2,000 proteins. But what is more outstanding is the quality of the data that is generated. We have much higher signal-to-noise ratio than whatever was published before, and we are even able to work without any carrier. So this is an adaptation of the SCOPE method. If for the one of you who knows the SCOPE method, in the SCOPE method, you typically use a carrier of 200 cells. In our experiment, you don't even need a carrier. And we can do the same with the label-free preparation. So we are currently collaborating with Bruker. We developed a new chip containing 48 wells for 48 single cell. And again, we developed a little adapter that allows us to interface directly with their liquid chromatography before injection in the timsTOF recipe. And with this method, we can detect up to about 1,000 and quantify up to 1,500 protein per single cell. We have the chance to be working with all the leaders in the field of single-cell proteomics. With Karl Merkler, we developed the multiplex chip. We are also collaborating with Ying Zhu at PNNL, Nikolai Slavov, who's one of the fathers of the field of single-cell proteomics, who developed the SCOPE MS, and some very nice researchers are Sinai Medical Center, Jennifer Van Eyck, and Stephen Carr at the Broad Institute in Boston. Thanks to all those nice research that we've done with those researchers, we were able to strike 2 co-marketing deals with both Thermo Fisher and Bruker. So we are promoting each other's system for single-cell proteomic analysis. And I will finish my presentation by a few slides on our latest device, the spheroONE, which, as I said at the beginning, is developed for sorting and isolation of spheroid and organoid. Why do we do that? Well, it's to allow you to obtain faster and better results while using those 3D cell models. This device allows you to sort those spheroids and organoids by size or by morphology or by fluorescent marker. By doing so, you are able to completely rethink the way you're preparing those samples. So up to now, most researchers working with spheroids will prepare their spheroids in those ULA microplates. So this is typically a plate that has been coated with a layer that prevents cell adhesion. You put about 1,000 or 2,000 cells in each of the well. You let them aggregate over a few days. Then you obtain a ball of cells, your spheroids. But by doing so, this generates typically a lot of debris in the wells. You get a lot of heterogeneity. Some well will have large spheroids, some well will have small spheroids. It takes a lot of space in your incubator because each 96 well plate will contain only 96 spheroids, obviously. What we offer with the spheroONE is a new approach. And we have also implemented a new mode of visualization compared to the CellenONE. We implemented the dark field imaging, which allows us to get very nice images of those particles. So you can do this by transmission. So this is kind of a bright field microscope. If here you have a spheroids. You can image them by dark fields, so you get the reflected light from the spheroids, or by fluorescence if you mark your spheroids with a marker. And what we propose with the spheroONE is to, rather than do one well, one spheroids, or one well, one organoids, which takes a lot of time and a lot of space in your incubator, here we think that a bulk spheroid formation is much more efficient. So you can use like what they call liquid overlay. So these are like, or you can use like micro well arrays, so like the L-plasia or the Kugelmeier plate. So these are plates containing wells, and at the bottom of the well, you have like multiple micro wells. This allows you to prepare hundreds of spheroids in a single well. Once you've prepared your spheroids, you then put them in suspension into the spheroONE, and then you start your isolation. You can then place them one in each of the target well, and you can sort. So you can make sure that you select only all the spheroids that contain the same feature, same size, or same markers, for example. So this allows you to have very high accuracy. So you can start from a bulk culture and sort, for example, for spheroids that are between 200 and 250 micron. Here we sorted between 350 and 500 micron. You can control the biomass. So you can put one spheroid, but if you want to put, let's say, 3 spheroids, 5 spheroids per well, you can also do that. By having a step of isolation between the formation of the spheroids and their assaying, you can get rid of all the debris. So you take your spheroid suspension, you put it on a strainer. This will remove all the debris, and then you start your isolation to obtain wells containing just a very nice spheroid without any debris around. This will facilitate a lot your imaging and improve the quality of your assays that you are doing downstream. And in the field of organoids, we've done some experiment with some brain pre-organoids, where we are able actually to select certain features of those pre-organoids in order to ensure that most of the pre-organoids that you're isolating will give rise to the right organoids afterwards. By being able to control the temperature in the target plates, we are also able to work with Matrigel, and we can embed the isolated spheroids directly into Matrigel. This is some example of our customer. We are very thankful to them because without them, obviously, we would not be there. We are working with many different cancer research centers around the world, single-cell proteomics center, some people working in microbiology for isolation of bacteria, sequencing platform, people working on rare cells. And we have some very nice partner and reference customer in South Korea, GI Innovation, the Korean Institute of Science and Technology, where we are able to undertake demonstration. If you would like to test our technologies, they have a CellenONE system. And we're also partnering with Green Cross Pharma, which is a major pharmaceutical company from South Korea. This is the team of my colleague. I'm very grateful for all the work that they are doing. We are a very international team. We have about 16 different nationalities, and we are a team of about 40 employees, very young and dynamic team. If you're looking for a job in Europe, well, don't hesitate to contact us. We are constantly looking for new talents to join our team. And I would like to also introduce our star in Asia. So Miss Minjung Jo, who's my colleague and senior manager for Asia. She's based near Seoul, and she's really great. And so if you have any question, any inquiry about our technology, don't hesitate to get in touch with Minjung. She knows the technology throughout and will be very able to answer most of your questions. That's it for me. I don't know if you have any question. I'll be pleased to listen to those. and to answer those, hopefully. Thank you.